So my name is Alexander Hefet. I'm a principal scientist at a company called Evotec. It's a British-German mainly company that are working in the field of drug discovery. I'm a computational chemist, and I work in this field for 15 years. And the subject of my talk today is Introduction to Computer-Aided Drug Design and GPCR Modeling. I hope that those terms will be more clear in the end of the presentation. So this type of presentation is slightly different from what you guys hear, which is more about the computers themselves. And in my session, it will be more about the use of the computers, what you can do with powerful computers in the fields of drug discovery. So there will be 4 items in the agenda. The first one will be drug discovery process in general, what it is the process, what is the stages of this process. The second will be, introduction to computer-aided drug discovery or drug design, and in shortcut it's called CADD, and application of CADD to GPCRs. This is a specific type of membrane-based receptors, which is one of the major targets for drug discovery. And I will bring a few examples of real drug discovery projects, so you will see in real how the computers was used. And the last subject will be biased ligands, which is also related to GPCRs. So if we're comparing classical and modern approach to drug discovery, so before we had the computers or before we had powerful computers, everything was done manually. So it was done by the chemist who was generating, synthesizing molecules, testing it against proteins, collecting the data, and analyzing this data with his brain, which is our brain definitely very sophisticated, but sometimes less powerful when it's coming to analyzing a lot of data. So usually it was based on the data that was produced for the project itself, without ability to compare it to large databases. And it was done mainly in the way of try and error. Let's try this and see how it works. Let's try this and see how it works. Usually this is very slow and inefficient and expensive process. When today we have computers, we are much more sophisticated, and we can do much more complicated stuff. One of the more complicated stuff, we can use the computers to analyze the data. And we have databases, and we can compare our project against large databases. In this way, we can provide what is called rationale and decrease amount of effort that we are putting in the projects. So rationale means speed, mean cost. And both methods practically, both approaches, its main target and main objective is to generate to predict the next generation of the compounds to make. In this first one, you will make a lot of compounds because you are trying different direction. In other method, you limiting amount of options, and this way you are coming to the target much quicker, hopefully. So when it all started when I was young. Finished PhD, coming to the company. I was quite lucky. I think my PhD was in the fields of computational chemistry. And I was lucky to start with a company, startup company, that was mainly based on computational chemistry, which is computational chemistry, which is slightly different from what you have in classical pharmaceutical companies, that they have huge number of medicinal chemists and small number of computational chemists that's supposed to support the effort of medicinal chemistry. Here was the approach philosophy was opposite. We have very limited resources of chemists. We have very limited resource of number of compounds that we can make. And when you have limited resources, you must compensate it with sophistication. You need to be sophisticated because there's limited amount of resources that you have. And if you look on how much compounds pharmaceutical companies made for each project, the numbers are thousands. They make thousands of compounds, even with all these sophisticated tools that we have. And they put a lot of compounds. And here you can see something very interesting. This was the compound number 23. So for the project, for all the projects, we made 53 compounds. The compound that reached the clinic, you see here the pills, that was produced for the clinic, and I'm happily smiling near them, this was compound number 23. So definitely computational chemistry can work, and computers can work, and not only virtual, but really produce real science. So drug discovery process, usually it contains 3 stages: heat identification, preclinical R&D, and clinical trials. Heat identification is to find the first molecule that binds your protein. And it's working the lock and key style. So you want to ignite your car, or you want to turn off your car, you need a key. This key is the drug. Lock is the protein. You put the key, you turn, your car is ignited. Something happening. The same thing working in drug discovery, in our body. You take a medicine, the medicine reaches the cell, find the right protein, ignite it, and something happen, and miracle happen, hopefully. Or opposite, other type of keys, you turn off. There's something happening in the cell, and you want to turn off this process. Again, you need a key. You put the key, turn off. Wonderful. So to find this first key, heat. This is what is called heat identification. I will talk a little bit about the methods, how to find the heat identification, but the result of this process is one molecule or series of molecules that can bind to your target protein. Second is preclinical R&D. This key usually is not good enough. It can be toxic. It can have other metabolic problems. It might usually bind weakly to your protein, and you want to do something with this. You want to change it. So this preclinical is actually to take heat, your original heat that you found, do something with it, change it, and go with it to humans. It's obviously contain many processes, including testing, including testing in animals. And you need to convince FDA, usually to give you a permission to work in humans. FDA is Federal Food Drug Agency, I think. This is yeah. Sorry? Administration, yes. Thank you. To give you a permission to try your new drug candidates in humans. In clinical trials, you also have a few stages. First stage, you're just trying on healthy volunteers just to see if the drug is not toxic, and then it goes to patients and try it in the beginning with a very limited amount of patients, and then for more patients. So this is the clinical trials. So there are the 3 basic stages. So just speak about the heat identification. The heat identification, the fact that you find the right molecule, the right protein is a miracle. It is. But there are the methods that doing it. So what you do actually, lock and key. So how you will find a lock? You will take a big tray with keys, and you will try to one by one to stack into your lock. Maybe you're lucky one will enter, and maybe if you're more lucky, you can maybe even turn it on the other side and open the door or other. This is practically what you do. So there are two ways to do that. Experimentally, take a robot that will do it one by one until, if you're lucky, you'll find some. Or you can do virtual. Virtual, you build in the computer, you build this lock, and you have structures of molecules, and you'll try by one to stack these molecules aside. In the end, you come with a short list. First step is expensive, very expensive, because you imagine how much experiments each attempt is experiment. If you have trying, for instance, Evotec have HTS library. This library of keys contain 400K molecules. So you need to perform 400,000 experiments. Now imagine that every experiment costs, I don't know, one euro at least. So you have 400K euro. When this is free, well, you just pay my salary. But computers run. They just do that. You come in with final list of compounds, with 100 compounds, and obviously you need to test them experimentally. So now instead of performing 400K experiments, you perform 100 experiments. You see the cost and the speed. This might take 6 months, that might take 1 month. So this is also example how the computers help science. When we are coming to next stage, preclinical, usually you find a compound that usually are in the range of 900 nanomolar, and you need to bring this compound to the range of 10 nanomolar to make it stronger. The fact that it's large number doesn't mean that you'll need a lot of material, a lot of keys to be able to bind to your compound, to your protein. Small number means that you don't need much. That means it's strong binder as it is. So the optimization of this compound is done in cycles, iterative cycle. Usually you start with analysis. You take what you see, you analyze, you're trying to understand, to rationalize how this compound binds to the key, what is the problems. Then you design a new molecules. You're trying to predict how, and I will talk about this a little bit more in details, techniques that we use to do that. But you design a new molecules and you predict that they will have a fixed qualities that you want to fix in analysis. Then you make them, the chemists make them, synthesize them physically, and we test them. And then again, we analyze the data. We're asking also, now, did we finish? Or we should, oh, sorry. Did we finish? Or shall we leave the loop and continue to the next stage? Or we should make more cycles? And this way it works. Usually in the industry, such cycle takes from 1 to 2 weeks. In the preclinical trials, again, we have 2 stages. So compounds coming from heat. This is our key. Wow, we found this key. Wonderful. Now we need to explore this key a little bit further to see what we can do. So we're trying to make a first attempt to improve the potency of this compound, to see if we can make it stronger. We perform very limited amount of optimizations. We're also comparing different keys, because usually you find more than one key. You're comparing, and you're trying to decide in this stage which is the most promising key that you can will continue intensively optimizing. Lead optimization is defined as the process of bringing the chemical series, the key, to the clinical trials, to iterative steps of design and testing. That's what you see, iterative steps. We are going into the cycles. There's a lot of debates about this, about heat to lead, because in old fashion, that was, you find a key, this key is bad, it's rusty, it's not working, it's not rotating, but you're still taking it and trying to do something with this, hoping that you will clean it with the time and optimizing it, and do something with this, and you will find something golden key in the end. The statistics showing that actually if you're starting with roasted key, with bad key, usually there's a high chance you finish with bad key. So there's a lot of questions regarding heat to lead, which key or which compound we should select. Should we start with something good, or we can have a compromise, or how much compromise we can agree. And in lead optimization, if we compare to heat to lead, we already trying to start with something good that have significantly potency to the target. However, there's other pharmacological properties that we need further optimization. So the fact that we have a key that rotate well and turn on our car, it's not enough. It must, we also have other properties like fit well in our pocket, for instance, and other pharmacological and metabolic approach. And we check in this stage also the selectivity, that it's not binding also to the other proteins, which might cause as a result not only wanted effect, but also non-wanted effect, which is side effect. Again, I practically spoke about this already. This is heat to lead. In the heat to lead, there's a lot of questions with. Is usually this is the stage that we checked these compounds, how promising it is. We're comparing with the compounds that was already known for this target and for other targets, and we see how much we can optimize it by making a few cycles to see if we manage to move from the original, from starting a potency to more potent compound. How much we can improve it, and evaluate how good this starting point is. And obviously then the next stage will be lead optimization. Again, you are familiar already with this cycle: design, make, test, and analysis. Obviously the computers are coming to analysis and design. So the first, obviously, in the analysis, we are usually the amount of data are growing. We want to compare this data to be able to predict what is next, what is the gaps in the data that we have, which compounds we can make. We usually started to make hybridization, to take two compounds that we made that was good. Maybe we can make some combination of them together, all the combinatorial, all these, and compare it with other properties, what is called SAR analysis. Structure activity relationship evaluation. So because the amount of data only for projects itself is huge, but you want to generate it, to compare this huge amount of data also with the larger data, comparing to the other projects, to the other molecules. And you need computer. We can't digest this amount of data. And to find some rationale, to find some trends in this data that will help us to design new molecules. Molecular modeling is obviously coming here when we are using this analysis and we build some models. Models obviously can have a different definition. It can be hypotheses that we have. It can be molecules, proteins that we build. It can be different ways, and I will extend this about the modeling in my second stage of my presentation. The next will be obviously making the compounds, testing these compounds. This is how the lead optimization works. Okay, introduction to computer-aided drug discovery, drug design. So, what is the definition of drug design or rational drug design? Is the inventive process of finding new medications based on available experimental data and molecular modeling techniques. So all the design is something that you do rational. You not try and error, you don't try everything. As my former boss said, if you want to try everything, you don't need computers. If you want to do rational way, the idea of to do something rational is to limit an amount of attempts only to those most promising attempts. Something that we are tend to believe or have a hypothesis that will work better than others. Computer-aided drug design is very simple. It's application of computer modeling techniques, programs to drug design process that often involve analyzing and filtering of the available experimental data, building some hypotheses, models. Again, the definition of models can be very extended. Models can be hypotheses, and models can be structures that we manage to build. But anyway, those tools that help us to predict and design new targets for synthesis. So based on those hypotheses, we design the next generation of molecules for synthesis. So this is hypothesis-driven drug discovery, or rational drug discovery, or design. So computer-aided drug discovery is actually divided to two major directions. One is called structure-based, other called ligand-based. In structure-based, it relies on the knowledge of a three-dimensional structure of the target protein and understanding how our ligand interact with our protein. So we need protein. In other words, we need the three-dimensional structure of our lock. So if you know how the lock look like, you can design a key for this lock. Sometimes it's available, sometimes not. We have other method called ligand-based. So you don't know anything about your protein, but you know you have some information on other ligands or other molecules that bind to the same protein. So what you can learn, what information you can get. So you can use methods to build hypotheses why those compounds that you know that they are bind to your protein, what they have. And then apply this to your molecule. So this is a completely different approach. So you don't know anything about target, but you know about other molecules. Structure-based and ligand-based. Just wanted to bring you a story here, very short story about the project that we had. For asthma, there was a protein, ITK, kinase, and we needed anti-inflammation drugs against kinase. And after we started with compound heat, our heat, HTS heat, there was HTS performed, this robot that puts, and he found that there's a molecule, there's a key molecule, 460 nanomolar compound. We was lucky, we had the protein of this, the structure of the protein. Here you have this is the protein, yeah? And this is the molecules. And we calculated the interactions between our ligand and this protein. And here every column here representing one interaction. So we had here 5 interactions. And the places that you have here is the residues that are in the binding site, the place where the protein bind our ligands, that don't have the interactions. But those are residues that are there, but they don't interact with our ligand. So the effort was to design a molecule, a new molecule, that will have to add some groups to decorate it in such a way that other residues that are there will also bind. So what we want, we want to generate to increase the attraction between our ligand and our protein. So in the end we finish which much larger number of interactions, and the attraction was increased by 100-fold. So it's 100-fold stronger. So yesterday there was Valentine's Day. So here they just boy and girl, and here is already marriage. So now they are already together. They are strongly bound with each other. In this method we have, but even if you have even if you have compounds, and we develop a method called FMO, which based on quantum mechanics, that you're not only showing you that in binary way that you have interaction or you don't have interaction, but it's really show you how strong this interaction is, because sometimes you have the interaction but you want to improve the strength of this interaction. Usually this is happening in the final stages of Lie optimization, that you want to make it very, very strong compounds, very strong binders, and here we manage to improve even slight changes of interaction. You can see here with the green stars. We increase additional to interaction, and again we manage to improve the potency by 10-fold. And this is real projects. So it looks simple, but this is how it is. So here you have protein, you can do a lot with it. So the question, from where you take the structure, the three-dimensional structure of the protein. So there is two experimental methods to do that. One is crystallography, when they manage to build a crystal structure of this, and then they solve three-dimensional structure, I mean the coordinates of the protein, and it's all stored in what is called protein data banks. You have website, it's all available. You have certain protein, just type and download. And what will happen if you don't have your protein? So then you can maybe model it. You can predict its structure. And I will speak now about this a little bit more in detail. So how you compare proteins? To compare proteins, usually you compare their sequence. They have amino acids, 20 amino acids. There's a certain order of those amino acids, and you compare them one to the other. That's called sequence alignment. There is a basic assumption that proteins, the proteins that have similar sequence probably will have similar structure, three-dimensional structure, if they have similar sequence. So if you don't have protein in database and you really want it and need it, you can try to build it with computer. The way to do it, you perform sequence alignment, you perform comparison of your sequence of your protein with the sequences of all the proteins that do have three-dimensional structure. And the cutoff usually more than 20%. So if you find something that have 20% similarity, This is your template. So what you do next? The next what you do is practically coming directly from the first stage. You already have sequence alignment. And what you do, you replace the residues of your target protein with, sorry, opposite. You replace the residues of your template with the residues of your protein, followed by optimization. Again, it's based on the assumption two proteins that have similar sequence have similar structure. So you just can replace one by one and convert the template into your protein. So suddenly you have protein. You use model instead of experimentalist. Usually it's a less good, but in industry compared to academia, you're allowed for a compromise. Because in the end, in why you can allow to yourself a compromise, because not the protein is the target. The protein that you build are the tools, only the hypothesis that you use for finding a new small molecule. The small molecules, you are very much in drug discovery, you are very much driven mainly by pragmatism. If certain hypotheses don't work, you throw it away. If certain protein don't work as a tool, you try another tool. If structure-based don't work, try ligand-based. You try everything. Okay, so you have protein, so what? You have protein, so what do you do with protein? What we care, we want to understand how our key working with our lock. We want to bring them together with this process called docking. Docking are computational protocols aimed at exploring the interaction between ligand and protein. We want to understand how our protein and our ligand interact with each other. So what we need to do, we need to take our protein, we need to take our ligand, bring them together. So this usually, what you need to do that? What you need to do? Sorry, how I optimize them? So you let the computer to do the work. You're taking the protein, you keep everything rigid, usually. There's other software that also the protein can move, but usually the classical way, you hold everything rigid in your protein and you rotate your ligand inside of the binding site, each trying different ways of binding. Each binding pose are scored, and in the end you have a list of different binding ways. And this is a lot of debates about this, which scoring function working well. Usually the way it's tested, again, the crystal structure where you have experimental data, and those scoring functions was built mainly on energetic terms and on empirical terms, tested how they were performed in finding crystal structure poses. But it's all coded. You have software today. There are many software that do this job for you. Again, it's usually built in two stages: protein, molecule, docking. First, you have search algorithm. That's what you saw in the movie. Molecule moved, rotated, so it's translated and rotated inside, and scored. And finally, it's all assessed, ranked, and you choose one. Again, since usually you have already some experimental data for your project, you see which of those poses can explain the data the best. So again, very empirical, very practical way. As you remember, we have a second method. The first method was based on the protein and understanding how the ligand binds to our protein. Other method is ligand-based method, which is based on the fact that I'm comparing my molecules against the molecules that can bind to this receptor. I don't know anything about the receptor, but I know about the molecules that bind to this receptor. So what you need to do, you need to be able to take two molecules and to align them one on top of the other. In this way, you can compare what is the difference. What you compare usually called pharmacophore. Pharmacophore is the groups, chemical groups on top of the ligand itself that usually responsible, or we think they are responsible for binding. So you look on two molecules that are together, you see one have one, so if you compare your molecule with this, you see one have aromatic ring and other don't have aromatic ring. So my molecules don't have aromatic ring. The molecules that do bind to the receptor do have aromatic ring. So assumption, let's try to add this aromatic ring to them and see what will happen. Again, it's all hypothesis. I don't know if it's for sure. If I have more molecules, I have 20 molecules, and all the 20 molecules that binds to my receptor do have aromatic group. That means, and my molecule don't have, so probably there is what is called consensus pharmacophore. That's probably something that are required, essential. And this is how the ligand-based work. You compare what one molecule have and my molecule don't have. Or sometimes my molecule have, or it's too big. And those other molecules are small. Maybe my binding site is too small and my molecules are too big and cannot enter. Sometimes you're improving things by removing what you don't want. So anyway, this method is based on ligands. Please don't ask me many questions regarding that because I just don't know. But to be able to perform all these computations, we have in Evotech, we have a platform. All the platforms are connected between different sites. And please, that's what we have. I was told it's impressive, but I don't know. Okay, so I think in this stage I will, before going to next two last items of the agenda, if you guys have any questions. Yes?
What is the level of confidence of these models for drug discovery that you just showed, or how they are validated?
The underlying question to that is, how sophisticated is the model that subtends all the results?
Again, there's no clear recipe regarding that. So for instance, if you're building homology model and your template is similar, have 80%, 90% similarity to your model, then you're more confident than it's 20%. The confidence is built usually are correlated with amount of data that you have, experimental data that your model is based on. But again, it's not score-based or anything. It's all experimental. And I think one of the maybe advantages that industry have over academia, that you can build a hypothesis and it's tested experimentally very quick. And it is, I think, the best test you have. So it's all very iterative. So your confidence are growing with those cycles that I showed. Yes?
What happens if you have a mutation of the ion channel where the protein binds, for example? Would you run like a whole bunch of simulations to see if your key is going to open that?
again, you need to be pragmatical. Obviously, you can do very, yeah, you try to incorporate the maximum amount of data you have. But you should remember one thing, the cycles are very quick. So whatever computational methods you do, they need to give the answer in a matter of days. So you can, okay, obviously you can try to dock into many states of the same receptor. But again, it very much depends on the project and the time that you have available to do those experiments. But what you mentioned, actually, it was called, I forgot the name of the program, concert docking, that the program docked automatically to many states of the same receptor, taking into account mutation and everything, if it's have effect on overall structure. Yeah? Any more questions? Okay. I would like to talk with you guys about family of receptors that are sitting on the membrane of the protein, called GPCRs. So what you can see here in the movie, they are sitting on the membrane. One side is facing outside of the membrane and to cross over the membrane, and it's finishing inside of the cell. Okay? The job of these receptors is to bind ligand from outside, change conformation. You see the ligands bind from outside. They are coming from outside of the cell, binds to the ligand. As a result, ligand changes forms, and you get signal into the cell. If you have excess of those ligands, or opposite, you have a lot of problems. Inflammation, and you saw them. Cancer, nervous system problems, and many others. So GPCRs, G-protein coupled receptors, are largest member of membrane proteins families. Almost every cellular process plus disease states controlled by its signaling to cell. GPCR enabled to cell to respond to external stimuli that you see, that activates or deactivates intracellular signaling. And between 40 to 60 of modern drugs are GPCR target. But I was going to tell you the history and how science changed quickly. So today there's nobody argue about the existence of GPCR. There's many GPCR structures that was solved crystallographically in the last, I think, 10 years. But in 1970, one of the famous pharmacologists in one of the papers said this statement. This would be true if I were so presumptuous as to believe that alpha and beta receptors really did exist. There are those that think so, and even propose to describe their intimate structure. To me, they are an abstract concept conceived to explain observed responses of tissues produced by chemicals of various structures. So 40 years ago, nobody believed that those receptors do exist as proteins. There was some assumption that there are certain mechanisms that ligands have effect on something that going into the cell, but nobody believed that there is a receptors. 40 years later, 60% of, there's between 60 and 40% of all prescribed drugs today are targeting GPCR. We know about 200 functional receptors that we know to which function in the cell they are connected. There's additional 600 that we don't know, and the drugs develop against only 50. So there's additional 750 receptors, and those 50 receptors are 60% of all drugs. So imagine how much potentially we still have with GPCRs. This is why GPCRs are so much in attention and the focus of drug discovery companies. Well, it's a not big secret that pharmaceutical companies is not only about saving human lives, they also care about finance. This was a little bit old prediction that by 2017 the GPCR drug markets will reach $120.5 billion. This didn't happen, but I think the result is 116. So there's a huge amount of money there. So everyone cares about GPCR, which is wonderful. That was even expressed in the fact that Nobel Prize for GPCR exploration was given to those two scientists, Professor Robert Lefkowitz from Duke University and Professor Brian Kubilka. Professor Robert Lefkowitz mainly was given for understanding the function of GPCR, and to Brian Kubilka it was awarded for solving the crystal structure of GPCR. So I hope I managed to convince you that GPCRs are important. The three-dimensional structure of GPCRs are growing tremendously. Many people, many groups are working on this. We have many GPCRs. GPCRs can be divided to three classes, class A, B, and C. Depends where the ligand binds and overall sequence. And I think this is one of my last slides regarding the theory and what is the GPCR there. There's a two in GPCR again, as I told you about the key and lock, we have activator and deactivators. We have the key that can turn on the car and the other key that can turn off. The activators cause agonist, they promoting activation as is a change, as you saw in the movie, change of the structure of the GPCR, and as a result you have binding of G proteins in from the intracellular part and causing a signal, or antagonists that actually lock the receptor in its inactive form and do not allow its activation. So agonist and antagonist. How we know this? We have practically crystal structures from different states, so we know the GPCR exists in multi-states, and we have experimental validation of the movie that you saw. So there's experimental, it's all experimentally validated. In Evotech, we develop a method to work with GPCR, although there's a lot of crystal structures. There's a lot of GPCR structures that their structure are not solved or not solved. It's not only when you saw that GPCR can exist in multi-states, so you need crystal structure per state, and this not exists. So you have an active state GPCR, but its active state completely different, and you don't have the crystal structure of this. So you need to be able to build it. So we have a method how to build this. In principle, it's not so different from what I was telling you before. Structure-based drug discovery, based on homology modeling. This homology modeling of GPCR, usually very similar to its template initially, the initial model. And GPCRs are not twins. You need to optimize it. You need to assign its unique features, and then we have a set of tools to optimize the structure and score them to increase our confidence. And those scores, obviously, most of them are based on comparing them onto the known GPCRs Sorry. Want to bring you examples here of a different case study for GPCRs. The project of finding a 5-HT2C agonist, serotonin agonist. The serotonins are receptors in our brain and also in our body, and for treatment of metabolic disorder. We run HTS. You remember heat identification. We run HTS. We found the compounds that we bind, all antagonists or partial antagonists. And we want we want agonists. We don't want antagonists. We want something that will activate, not deactivate. So what we should do? This was the starting point. So what we did, we built active and inactive states of this receptor using homology modeling. So we have now our receptor in two states. We docked our ligand, the heat that we found from heat identification, in both of them, and so what is the difference? Why our ligand prefer to bind to inactive state instead of active? We compare, this is what you see here is comparison between active and inactive. Red is where they're different, green is where the same. We are talking about the same receptor during active and inactive state. We know that GPCR in their resting state, usually in our body, everything is resting until it's stimulated. Otherwise, we'll be all the time like an Energizer bunny, all the time moving. So we are not, everything is sleeping until it's stimulated. So that means that inactive states of proteins in our body are energetically favored. When the ligand that activate, it's actually stabilize the active state. It's giving the extra energy to stabilize the active state. That's what how agonist. We looked, I will not go too much into details, and practically we understood that our ligand cannot compensate enough of loss of energy of active state. If we want to convert it to agonist, we need to increase amount of interactions. But for only very specific interaction that we managed to find by comparison active and inactive state. And as a result, we managed to find molecules that was, you see there's no much big difference, but we managed to find molecules, this was starting, this was end, that was very strong, agonist. Again, as you see, the first process, without going into technical details, but you need to, first of all, you after a rationale. You need to rationalize your data and then design the molecules that should address the problem, hoping that it will work. Sometimes it works. Again, additional project. HTS heat gave us huge molecule. Sorry, the story was a little bit different. There was a company in Australia, Alchemia. They have a library of some molecules are natural molecules are big peptides. It's very hard to compensate peptide with drug-like, drug-like molecule. It's very hard to do that, very, very challenging. So they found out that there is a small molecules that are based on with a sugar in the center, that have three vectors, that those small molecules that look like more like small molecules than compared to peptide, they can replace peptide. And they ask us what we could, okay, but this is too big to be a drug. What we can do with this? And then we combine ligand-based and structure-based to understand this, and sorry, I'm just rushing a little bit because of my time. We managed to find the molecules that look more like drug-like molecules, starting from this. I'm going to skip this. We had additional project that I wanted to speak today, but I'm going to skip this, and I'll go to the slide just for your information about the design. That's actually the golden goal of computational drug discovery. When you have ligand and you understand how it interacts with your protein. Imagine, and then the result is, in red, you tell to the chemist, look, green, this is where you can change. Red, don't touch. Because if you touch, you will lose. Imagine you don't have this. Think about number of options that the chemist must. So all these places, he will start to work, he will change here, it will spoil, maybe, okay, maybe I make this molecule shorter, maybe I do here. Amount of effort will be huge. Amount of combinations here will be huge. So this thing provides the rationale. This is what is all rationale all about. To tell where you can change or should change and where you can't change, where you shouldn't. I think this is the key message. Okay, I'll skip this. Just give you something that recently discovered. Recently discovered, everyone believed in all these years that GPCRs mainly activate or send the signal Y via G protein binding. Apparently, it has alternative binding, alternative signal. So our GPCRs can send, instead of one signal, it can send two signals to the cell, which other signal was not very familiar. What the meaning of this? The meaning of this, that one will give you a benefit and other will give you side effect. So in many projects, there was a known, if you take a drug, usually inside of the box you have instructions and a huge list of side effects. It might have that and that and that and that, and sometimes there is no clear indication what causing this side effect. The belief was that the medication go and bind some other protein. And these other proteins that are not known because we don't know, we done everything, we can test every protein in the cell, but it's doing something, somewhere. In case of GPCR, it was discovered that the same receptor is double agent. It's doing one good thing, another spoiling. And I'll show you how the practical of this. I'll just skip all this. Just give you a few examples. There's a company. Again, the moment something nice discovered, there's always some spin-up company that trying to use this, which is great. Morphine. You guys know. Morphine, drug, analgesia, painkiller, yeah? There was a known that the dosage of morphine that you can give are limited because there's two major side effects. Constipation, which is relatively less problematic, respiration, depression. So you can't give morphine in huge dosages because the patient will suffocate. Many years people didn't understand from where it's coming. Apparently, it's coming because of the second signal. So if you make a compound that will have only one signal, what is called biased ligand, that have bias towards only one signaling pathway, you can have a drug that will be a huge painkiller without this side effect. So it's very practical. It's not, it's not theoretical things. Additional example. Again, angiotensin receptor, known for controlling blood pressure. One signal causing increase of blood pressure. Other signal cause totally opposite, decrease. So if you manage to separate those two signals and signal only one, you will have all these positive effects. So people developed drugs against angiotensin receptor, but didn't understood how it even work because of this double signaling. So this something that are completely new and something that we still don't have computational technologies how to generate those biased ligands that will have only one signal. The last thing that I wanted to show to you guys is our collaboration with academia. I think academia have a huge advantage. It can do proper research instead of biased research that we have in industry, because it's all very much project related. And this is a huge opportunity for us to work with the group, a privilege to work with the group of Professor Anders Thorson Nicholson. We are working supported by ComputerAided, supported by BBSLC, where I'm trying to provide the computational methods, and Andreas' group working on experimental methods. We have shared student, which is very interesting experiment, very experimental in terms of studentship. Usually before you had always computational chemists, you have experimentalists, and Andrew is kind of hybrid. He knows from both sides. He was also working lab and also working computer, which is amazing. So we are working mainly on residence time. So it is known not only the molecules, effect of the molecule, a therapeutic effect of the molecule, very much depends how long the molecule sits in the binding site. More it sits, more effect you have. And we are working on that. It's not always related to attraction. And recently my book came out, in January, Computational Methods for GPCR Drug Discovery, and you can find a lot of methods, a lot of strategies to deal with to use the computers in the fields of GPCR drug discovery. Thank you guys, and I have a bunch of people who will help me a lot. Dmitry Fedorov from FMO developing, my wonderful team on Evotech, and Andrea for our collaboration, and ComputerAided for bringing me here today. And also other things, of course. Thank you guys. If you have any questions.